What a plant sees. This videotape will be the first chapter in a charming book just published last fall by Daniel Chaimovitz called What a Plant Knows, a field guide to the senses. He is a plant physiologist in Tel Aviv, but he has written this very interesting book aimed at less advanced levels of understanding of plants. And so chapter 1 is What a Plant Sees. And they're watching us, it turns out. Plants see you if you're near them. They know if you're wearing red or blue. They know if they're in a new window. They know when spring is coming. They know if it's morning or evening. And actually quite a lot of other stuff we probably don't even realize they know. They do. So what a plant sees. And this is a quote from Daniel. So if you take someone who's completely blind and by surgery in some way give them a camera allowing them to see just shadows, we would say that that person now has rudimentary sight. He doesn't see pictures, but for that person being able to differentiate shadows is definitely sight. If we would let them then be able to differentiate between red and blue, that would be even slightly more sight. Well, that's what plants do. They don't see pictures, but they see colors, and they see directions, and they see intensities. And on a certain level, plants might think that we're visually limited because plants see things that we can't see. They can see UV light, and they can see far red light, and we can't see those at all. So I think that we can say that plants see. A plant knows quite a bit, much more than we give them credit for. To review a little bit before we go further, just for some terminology, the electromagnetic spectrum, which is a gradient of wavelengths of increasing or decreasing energy, depending which way you're coming or going. The longest or the longer that we commonly deal with, slower wavelength is radio. AM radio can be up to half a mile long. Then gradually we move up to microwave with higher intensity and shorter wavelengths, and then finally get to heat, which is infrared. And then we get to this tiny little bit that we can actually see with our eyes, which starts with far red, and then red, orange, yellow, green, blue, purple, and then ultraviolet, which again then we can't see, which is higher energy than any of those that we can see. And then finally X-rays and gamma rays. And what I'll be talking about here is constrained to the far red to the ultraviolet. And then our eyes, since we're talking sight, on the lower right you can see a diagram showing light coming into the eye, and then it bounces on the back of the eye on the retina. And then I'm sure you've all heard before, we have rods and cones, little funny structures, that are contain different kinds of molecules called photoreceptors that absorb different wavelengths. And so you'll have a green photoreceptor that will absorb light that's in the green wavelength ranges, and the same for blue and red and purple, but not ultraviolet because we can't detect ultraviolet. People that are colorblind, if you're red colorblind, then you don't have the functional red receptors in there. So some people even see less than what is indicated. And in the upper left is just a little bit more detailed diagram of the eye, and showing you that it is the back of the retina where these special structures are. And according to the information provided, your eye is roughly equivalent to a 130 megapixel camera. So they're pretty concentrated. A lot of digital cameras are 8 at best megapixels. So how do plants see light? Obviously they don't have a retina and everything like we do. So however, it's known that essentially all plants will bend toward a light source, and so they can see the light. They know there's a light on one side of them and not on the other. And even Darwin, the guy that first formulated thoughts about evolution, was fascinated by this fact, and I'll show you an experiment he did on the next slide. This response is called phototropism, and it turns out it's a reaction to blue light, not other colors. So if you shine a red light or UV at your plant, it's not going to respond for you. It's just blue light. And the other interesting point is that bending occurs very close to the growing tips. So here's an experiment that Darwin and his brother did back in 1880, excuse me, and their experiment is the 5 little seedling drawings on the left. The other 2 are a second experiment done later. And what he did was show that he could make plants go blind. These little seedlings have light coming from one direction, The first seedling represents a control that he's done nothing to it, just so that it represents, the conditions were such that if the plants could bend, they would. Then he had seedlings that he cut the very tip off, and it turned out those plants went blind. They did not bend toward the light, even though there was plenty of plant left. And then he could cover the tip with an opaque cap, a little piece of foil or lead or something. And those plants did not bend. They became blind. But if he covered them with a tiny little piece of glass, a little cap of glass, they would still bend. So that showed that it was not just the fact that there was something on top of the tip. It was that if light could still get to the tip, then the response could occur. And then finally, just as an additional control, he put an opaque band around the bottom of the plant to see if somehow the lower level of the plant was responsible for seeing. And that was not the case. So the plant without the tip or the covered up tip went blind. The plant with a covered base or a glass cover on the tip could still see just as well as the control. And then some years after that, two gentlemen, Boyson and Jensen, extended the experiment by cutting the tip off, but then putting a tiny little slice of agar between the tip and the plant. And agar, like in a petri dish, which is porous. And then as an alternate treatment, they put a little piece of mica, which is impermeable substance, in between the tip and the lower bit of the plant. And sure enough, if it had a permeable substance there, the plant was not blind. It could bend. And many years later now, we know that there is a hormone that comes out of that tip and oozes down, is transported down to the lower sections of the plant. And the area closer to the sun gets less hormone, and so those cells don't grow, and the cells farther from the sun grow more because they receive more hormone. So that's how the plant is seeing and responding to the light. Okay, another way to look at how plants can see is looking at long day and short day plants. There are plants that don't bloom until a certain length of daylight occurs, and so these are the things that you think of in blooming May and June, when the days are getting longer. And then there are short day plants that bloom in the fall, or later than June at any rate. Our longest day is generally the 21st of June, so any time after that, days are getting shorter. But so if you think about the plants that bloom in, chrysanthemums and goldenrods, those would be called short day plants because they don't bloom until the days are getting significantly shorter. Well, somebody figured out that if you hit them with a flash of light, not even for very long, a minute or less at night, the long day plants will bloom, even if the day is not as long as they normally would wait until they begin blooming. Flashlight at night, boom, they start blooming. But then the short day plants, if you give them a flash of light at night, then they don't bloom even if the days are short enough that they normally would be triggered. And so what people realize that it's not the length of the day, it's the length of the night that makes a difference to these plants. So they really aren't long day plants, they're short night plants. And by giving them the flash of light at night, you've, regardless of how long the night was going to be, you've roughly cut it in half, or at least lopped a significant amount of time off it, so they think they've had a short night, and they bloom. Then on the other ones, the ones that we call short day, that are actually long night plants, if you give them a flash at night, then they're convinced their nights are not long enough, and they don't bloom. So it's the length of darkness that these plants are measuring. But then, of course, the question is, how are they measuring it? How do they know? So what are they seeing? Well, the next discovery was that red light is what causes this effect. You get a weak or zero response if you use other colors. And that if you follow the red light treatment with a far red light treatment, so a longer wavelength, getting closer into actually infrared, then that cancels a response. So if you've given them a flash of red light, and so now they're going to decide their nights have become dramatically shorter, then you follow that with a far red, and they go back to not thinking their night had been interrupted. And you can toggle back and forth, far red, red, far red, red. And they always remember the last color they saw. So they always respond to the last color they saw. So in nature, this kind of makes sense. You think of the last light at night is going to be the longest red wavelength. As the sun goes down, the wavelengths are getting longer and longer and longer. And so the last thing they see at night is far red light. And then at dawn is the reverse. First you're going to see far red, and then shorter red light. So sun's come up, red light, sun's gone down, far red. So how do they see these far reds and reds? Leaves are required. It was known a long time ago that if you shine the light on just one leaf, you'll get your response. But if you remove all the leaves, you don't get a response. So somehow something in those leaves is being used for timekeeping effectively. And so nothing much could be done just because of the difficulties of experimentation. But if you recall when I talked about the Brassicaceae, there is a plant called Arabidopsis that has been used as a workhorse in plant genetics and biochemistry laboratories for many, many years now. It's called the fruit fly of the plant world. And so sure enough, people used Arabidopsis to generate mutants that didn't respond to light like normal plants. And they came up with a lot. There were plants that, just a spectrum of different responses. And this would be an example of some of the things. Here across the top row, you've got plants that are not mutants. So WT, they're wild type. And then the next row is one type of mutant, and the next row is another type of mutant. And then the left column is the control, so those plants have gotten normal light. And then the other columns, they've gotten different light treatments. And you can see all the different responses they've had. Their leaves are different, narrow, skinny little leaves in one case, and broad leaves in another, and starting to bloom and not starting to bloom. These treatments are end of day, far red, high intensity red and far red, and low intensity red and far red. So these types of experiments allowed them to, scientists around the world, in dozens and dozens of experiments, eventually figured out that there are photoreceptors in these plants that are responsible for detecting these different types and qualities of light. And they're called phytochromes or phototropins. There's a few other types. And there's actually about 11 in Arabidopsis, and which is interesting because there's really only 4 in humans. We have one set to see dark or bright or dim, black or white basically, and then 3 main colors, red, green, and red, yellow, and blue. So light, this is one way of underscoring how important light is to plants. They can't, after all, move, and plants are their source of food. Light is their source of food. They're going to use light to photosynthesize. So it's important to them to optimize their response to the quality of light they have. And so in all, these photoreceptors combine to tell the plants when to germinate, when to bend, when to flower. If it's nighttime, if it's really bright or really dim, maybe another plant has grown over you, and now you need to figure out what to do and even help it keep time. So the way these phytochromes work, just in a general sense, is that it is a protein connected to a compound that is a dye that's sensitive to light, and is in one form under one kind of light and another form in another kind of light. So in the morning, red light comes in and hits the plant, and all day long this phytochrome is converted to the red light activated form. And then as night goes down, as the sun goes down and far red light hits the plant, then the far red activated form it comes in, and then slowly converts during night, it slowly converts back to the red activated form. And so the concentration of which state of this protein dye combination is in tells the plant how long it's been dark or how long it's been light. So, responding to light information then. It's not enough to merely detect the light, the plant must respond. And this effectively is sight, interpreting the data. Just like we see things with our eyes, and our eyes give our brain information, and then our body responds. Whether we have a chosen response or it's a physiological response, just like your skin tans in the light, or you respond to the day length when the time changes or something. So animals have a similar need to collect information, surroundings, and respond. We just have a very different, or not really a very different system. We have a different system. So our red light photoreceptors are not the same as those in plants, but they are similar in that they both are a protein connected to a dye, and they absorb light and convert to a different form. And interestingly then, to wrap this up, our blue light receptors, which are called cryptochromes, are very similar to plants, and these are like our internal clock, and that regulate your circadian rhythms. You know, if you travel far enough that your the day-night cycle is very different than what you're used to, you get jet lagged. And plants, and that's because your body is responding to the cryptochrome data collected from the blue light receptors that are telling you it should be morning, it should be night, you should be eating. Just all that internal clock type of mechanisms. And then plants have similar processes, like open your flower in the morning or open your flower at night when the moths are out, or whatever. And those receptors are so primordial that they're shared amongst humans and animals and mice and insects and even some bacteria. So that concludes how plants see.